How can we understand the way the nervous system and the body work together to generate behavior? The human brain has about 10 billion neurons, and the human body is very complicated, so it may be useful to look at a somewhat simpler animal to answer this question. Dr. Hillel Cheel's lab studies the neural circuits, body, and behavior in the sea slug Aplysia californica. Understanding how the brain and body work together could lead to better brain-computer interfaces to control smart prosthetic devices like an artificial hand. It could also help design soft robots with flexible grippers that can handle slippery materials like seaweed. When a slug finds seaweed, it first tries to grasp it. This is called biting. The slug is using its tongue-like structure, the radula, to try to grasp seaweed. Once the slug succeeds in grasping seaweed, it begins to swallow it. How does the slug swallow? It uses its feeding apparatus, the buccal mass, which is controlled by the neurons in the buccal ganglia. Let's go through a swallow cycle and see the muscles and neurons that are involved. There are two main movements that make up a swallow cycle: opening and closing the grasper, and pushing it forwards, protracting it, and backwards, retracting it. Let's start by looking at opening and closing. The surface of the grasper is a thin sheet of cartilaginous-like material covered with backward-facing teeth. It is called the radula, which is the Latin word for rasper. The radula curves around a very large muscle, the horseshoe-shaped I4 muscle, which is stiffened by cartilage. When the I4 muscle contracts, it pulls the halves of the surface of the radula together. This closes the grasper. The B8AB neurons cause the I4 muscle to contract and thus control closing of the grasper. The grasper is opened by small muscles. The subradular fibers and the strap-like paired I7 muscles are activated to cause the grasper to open. The I7 muscles are activated by the B49 motor neurons. Now that we understand how opening and closing work, let's understand how the graspers move forward and backwards. A thin hemispherical muscle that wraps around the back of the buccal mass, the I2 muscle, contracts and pushes the grasper forward towards the jaws. This movement is called protraction. The I2 muscle is controlled by the motor neurons B61 and B62 and the multi-action neurons B31 and B32. Retraction is controlled by the I1-I3 jaw muscle complex. When it contracts, the grasper is pushed backwards towards the esophagus. This is known as retraction. Neurons B6, B9, and B3 generate the main force in muscles to cause retraction. Now that we understand how all the different parts work, let's see the entire process of swallowing. First, the grasper protracts due to the I2 muscle, and it is open as it protracts due to the I7 muscles and subradular fibers. Second, the grasper closes due to the I4 muscle near the peak of protraction grasping the seaweed. Third, the grasper retracts due to the I1-I3 jaw muscle pulling seaweed further inwards. The slug can adjust its behavior depending on what is happening in the environment. If the seaweed is hard to eat, it can activate its neurons more strongly and for longer so that it can generate more force and break off a piece of seaweed. Studying the neural circuits and the body of the slug show how an animal can adjust its behavior from instant to instant and provides insights into how we adjust our own bodies as the world changes around us.